get started. Good to see everybody. So it's Friday, so you'll get your quiz at the end of the lecture today, and it's going to be on the genes chapter, all those concepts that we went over on the previous chapter, but we've now started a new one on viruses and prokaryotes. Let's see, there's one more midterm left in this class before the final exam, and it's not next week, but it's the week after next. And so next week when we start meeting, I'll tell you exactly what topics are going to be on that particular midterm. All right, well, let's get back into this lecture. So the chapter is on
and then she went out the other door.
The chapter is on viruses and prokaryotes, and I thought I'd start off on just with the viruses. Let's turn down the lights a bit. Viruses, remember, are extremely simple organisms. All they are is a very small chromosome with just a few genes, somewhere on the order of 10 or 20 or up to 100 or so, and a protein coat called the capsid, which surrounds and protects the chromosome. And that's it. No organelles, usually no membrane at all, no active metabolism, that is no enzymes inside them carrying out any chemical reactions. That's all they are. And they're a lot smaller than cells, even smaller than prokaryotic cells. And they're so small and simple that not all biologists consider them even to be alive. And in fact, they can't even reproduce by themselves. The only way they reproduce is to get inside a living cell, an actual cell, and then trick that cell into making copies of the virus. And that's what this yellow line is supposed to be right here. This is some cell, and the virus has now gotten inside of it. And I put this membrane protein here to show that viruses use the membrane proteins as gateways to get into the host cell. These membrane proteins, like receptors and membrane transport proteins, always have a binding site where they're supposed to bind to their regular ligand molecule, but part of the capsid proteins masquerade as the ligand, bind to the binding site, and by attaching to it like that helps them get inside the cell. Anyhow, once the virus has gotten inside the cell, it now has to trick the cell into making copies. And usually this ends up being fatal for the cell, meaning that when the cell starts reproducing the virus, the cell will make so many viruses, the cell will become filled with too many viruses, and then like an overinflated water balloon just explodes, and that releases all the offspring viruses, which then infect neighboring cells, and that's the way the infection spreads. What we're going to do now is essentially look at the viral life cycles, study exactly what the viruses do once they get inside the host cell and how that leads to the host cell reproducing the viruses. I'll do something with that. There's actually two slightly different methods of reproduction that the viruses can use once they get inside the host cell. One is called the lytic cycle, and one is called the lysogenic cycle. And in a nutshell, the lytic cycle is where the virus gets the cell to actively and very quickly reproduce the virus, and so this quickly leads to the death of the host cell. Whereas in the lysogenic cycle, it's more like the virus hibernates inside the cell for a while. All right, let's do the lytic cycle first. Now, so it is the viral reproduction pathway where the cell actively reproduces the virus, usually so much so that the cell bursts. To see how this lytic cycle functions, we have to sort of subdivide what we're talking about into RNA viruses and DNA viruses, because with some viruses, the chromosome is made out of DNA, just like our chromosomes are. But in other viruses, the chromosome can be made out of RNA, which is pretty weird because all other life forms on Earth, prokaryotes or eukaryotes, you name it, all other life forms on Earth use DNA as their genetic molecule, but some of the viruses do have RNA as their chromosome. But in any event, let's start off talking about the DNA viruses and see how they do the lytic cycle. Okay, so the virus enters the cell, and for the DNA viruses, essentially what happens is this, that the DNA chromosome of the virus pops out of the capsid, it exits the capsid, and to the host cell, this is just going to be another chromosome. You know, cells can't count how many chromosomes they're supposed to have inside themselves, and so the cell just thinks, oh, here's another piece of double-stranded DNA, that must be a chromosome. And so it does the same things to the chromosome, like express the genes and duplicate the chromosome, that it does to its own chromosomes. And the way I want to present this is to talk about the enzymes that are involved in viral reproduction. And some of these enzymes are host enzymes, but some of these are not. Some of these are going to be viral enzymes. All right, as far as host enzymes are concerned, one of them that gets involved here with the DNA viruses is good old RNA polymerase II. And remember what RNA polymerase II enzyme does. It binds to promoters and then makes a messenger RNA from the gene that follows that promoter. We just did that in laboratory on Wednesday. And so that's what happens here. The viral chromosome has promoters that are recognized by the transcription factors and RNA polymerase II. And just to give you an example of what one of these viral genes is, remember that the capsid is made out of proteins, and so there have to be genes on the viral chromosome that encode those capsid proteins. So let's use that as one of the examples. Anyways, there's a promoter for that viral gene, and here's the RNA polymerase II. It finds that, well, goes down there, latches onto the template strand on the viral chromosome, and cranks out a complementary RNA strand, which is the messenger RNA. So, for RNA polymerase II enzyme, the template that it binds to, that the RNA polymerase II binds to, is the viral DNA chromosome. And the product of what you get when that happens is an RNA that's complementary to one of those strands. And in this case, what I'm talking about is, of course, the messenger RNA. But I'm going to write it slightly funny. I'm going to write a cRNA. And the little c stands for complementary. And that is what it is. The messenger RNA is always complementary to one of the strands on the DNA. All right, so there's that messenger RNA for the viral capsid. And then what happens next is pretty familiar. A ribosome will now bind onto that messenger RNA. Let's see, there's the mRNA. A ribosome binds onto that messenger RNA just as if it was a messenger RNA that came from one of the host cell genes, because the ribosomes don't know that this messenger RNA came from a viral gene. And then the ribosomes translate it into proteins. That's where the new capsids come from. Now I don't think I have the ribosomes in this cartoon, but imagine the ribosomes are there. They translate that, and that's where the new capsid proteins come from, like that. So far the virus, the DNA virus, has partially gotten the cell to reproduce itself. It's got, just by having the cell express these capsid genes, the cell has made some new capsids for the virus. But to be a complete virus, we have to have copies of the chromosome to go inside each of those capsids that are made. And again, the host provides the enzymes from that. Oh, I should clarify here. When I say it makes a complementary mRNA, functionally, that is the messenger RNA for genes like the capsid proteins. Okay, but anyway, the next step is that the virus has to have the cell make copies of the chromosome, but that's easy because the cell does that naturally with its own chromosomes, is make copies of it. What enzyme in the host cell is used to copy chromosomes, to duplicate chromosomes? Anybody know? Think back to when we talked about chromosomes getting duplicated. That's very, very good. Yeah, there's that host enzyme, DNA polymerase III. And remember, that's the one that uses primers and ligase and things like that. But the point is, this DNA polymerase III can bind onto one of the strands of the chromosome and make a complementary strand. And so eventually, the two strands of the original chromosome each get complementary strands, so you end up with a duplicated chromosome, copies of the chromosome. And what happens is the cell does that to the viral DNA chromosome. So you get complementary DNAs to the viral chromosome, to each strand of the viral chromosome, and this ends up making duplicates of the chromosome. And that's what we're going to see here. There's the DNA polymerase III. It says, oh, there's a chromosome that I need to duplicate. And so it makes us a complementary DNA of each of the two strands, and that ends up making a copy of the virus, the viral chromosome. And then those load inside the capsids, like that, and now the cell has reproduced the virus. And so eventually, the cell just explodes from all the viruses it makes, and then those drift out and infect neighboring cells.
So the virus chromosome has all the primers and all the motors?
The genes on the viral chromosome definitely have promoters, and so that's how they're able to fool the cell into binding on with the cell's RNA polymerase II and making messenger RNAs from those. And the viral chromosome also has sequences, just like the host chromosomes do, to attract the DNA polymerase III and ligase and primase and all the enzymes that are used to duplicate chromosomes. Yeah, so I guess the shorter answer is the viral chromosome has all the sequences it needs to fool the host cell into treating it like a regular old chromosome. Kind of sly of the virus, you might think of it that way.
So then it's because there is a generalization between all of the different chromosomes of different cells that have, so they all have the same kind of motors.
Well, yeah, because you mean different cells between different species?
Yes.
Well, yes and no. I mean, the sequences that you find in human genes for our promoters and our chromosomes to get duplicated are not necessarily the same ones you would find in plant genes. And that's part of what makes viruses specific to generally just a few species and not generally to, across species. Okay, so all this was how DNA viruses go through the lytic cycle. And notice for DNA viruses, they don't really need to carry any of their own enzymes. All the host enzymes, all they need are the host enzymes because their chromosome is DNA, just like the host chromosome. For those viruses that have RNA chromosomes, it's kind of a different matter because the host cell is not used to having an RNA chromosome. That's not something that occurs naturally in organisms. We have DNA chromosomes. And so the viruses usually have to provide an enzyme or two to help the host cell express their viral genes because the host just doesn't have the enzymes necessary to express genes from RNA chromosomes and to replicate those chromosomes. And I should mention here that a surprising number of viruses are RNA viruses. You would think that the DNA viruses would have the advantage because they're very similar to the host chromosomes. But for some reason, it's just the opposite. Most viruses that affect human beings and other species are actually